Hey guys, this is Jason Matthew from Trinidad and Tobago, and today we're going to be answering the question, how many ATP can be generated from one molecule of glucose? There's another very important question we have to ask. Is oxygen present? And if it's an eukaryotic cell, do we have mitochondria? Now, if the answer is yes, then we're looking at aerobic respiration. If the answer is no, we're looking at anaerobic respiration. So for aerobic respiration, if you look in the different textbooks, you're going to get a range from 30 to 38. Alright, so let's see how they got those numbers. So first of all, for aerobic respiration, we're looking at 4 metabolic processes: glycolysis, the link reaction, which is the reaction catalyzed by pyruvate dehydrogenase complex, the Krebs cycle, otherwise known as the TCA cycle or the citric acid cycle, and then oxidative phosphorylation. For glycolysis, we are converting 2 molecules of glucose to 2 pyruvate molecules, and in so doing, you're going to get a net gain of 2 ATP molecules and 2 NADH. Now that pyruvate is made in the cytosol, so the pyruvate will then enter the mitochondria where it undergoes the link reaction. So 2 pyruvate is being converted to 2 acetyl-CoA molecules. And we call that the link reaction because we're going to be linking both glycolysis to the TCA cycle. Now when these 2 pyruvates are converted to 2 acetyl-CoA molecules, you get 2 NADH being produced. Now each of these acetyl-CoA molecules, so each one, is going to react with oxaloacetate to form citrate, and then citrate will go through a series of reactions to reform oxaloacetate. We call that one turn in the Krebs cycle. Alright, and that's for each acetyl-CoA molecule. And for every one turn of the Krebs cycle, you get 3 NADH, 1 FADH2, and 1 ATP. Since we have produced 2 acetyl-CoA, it means that we're looking at 2 turns of the Krebs cycle. So it's 2 times 3 NADH, 2 times 1 FADH2, and 2 times 1 ATP. Now all that NADH and FADH2, they will then be diverted to the oxidative phosphorylation, which includes the electron transport chain and ATP synthase. Let's summarize what we have covered so far. So for glycolysis, we have 2 ATPs being generated, 2 NADH, well, I mean a net gain of ATP, I should say. No FADH2. For the link reaction, we are only getting 2 NADH. For the Krebs cycle, 2 ATP, 6 NADH, 2 FADH2. So now we're down to the 4th metabolic process, which is oxidative phosphorylation. First, we need to make the assumption that 1 NADH will give you 3 ATP, and 1 FADH2 will give you 2 ATP. Now, let's look at first the amount of NADH that we have here. 6 plus 2 plus 2, that's 10 NADH. So 10 times 3, 30 ATP. Then we do it for the same thing for FADH2. We have 2 FADH2. So 2 times 2, that's 4 ATP. So it means that for oxidative phosphorylation, you are getting 34 ATP. And if you add up all the ATP molecules that we have there, that will give you a net gain of 38 ATP. 2 plus 2 plus the 34. So that's 38 ATP. So we have achieved, or we now understand how we get 38 ATP. Alright, but as I said, there's a range. Some textbooks will say 30, 32. So let's see how we get those numbers. Now to get those numbers, we have to go back to the assumption. When we did experiments and so on, we realized that 1 NADH will really give you 2.5 ATP, and 1 FADH2 will give you 1.5 ATP. Now that might sound strange at first, 2.5, 1.5, why not whole numbers? Well, to answer that question, we have to look and see what's happening inside of the mitochondria. So let's go there, guys. So this is inside the mitochondria here. This is the inner membrane of the mitochondria. We have the intermembrane space, and what is not drawn, but if we was to draw it, we will have the outer membrane somewhere here. And then this is the matrix or inside of the mitochondria here. Now, spanning or inside of the inner membrane, we have the electron transport chain. Now the electron transport chain comprises of 4 complexes. Complex 1, 2, 3, and 4. Now complexes 1, 3, and 4, they span the inner membrane. Complex 2 is embedded in the inner membrane. And on a side note, complex 2 is really succinate dehydrogenase complex, which is also part of the TCA cycle. Now, as electrons move from each complex to the other, complexes 1, 3, and 4 play a very important role. They pump protons from the matrix to the intermembrane space. Alright, now the numbers are very important. Complex 1 pumps 4 protons, complex 3 pumps 4 protons, but complex 4 pumps 2 protons. And from experimentation, we saw that, or we observed that as 4 protons, it takes 4 protons to pass through ATP synthase to produce 1 ATP molecule. So as 4 protons passes through ATP synthase, you get ADP plus Pi. In other words, ADP is being phosphorylated to 1 ATP molecule. Now these numbers are very important. Now another thing that we have to talk about is what's happening with the NADH and the FADH2 that we were talking about before. Now NADH will give up its pair of electrons to complex 1. So it means that NADH will, by giving up its electrons to complex 1, as much as 4 plus 2, that's 10 protons, will be moving from the matrix to the intermembrane space. Now let's do some mathematics now. We just said that 4 protons will give you 1 ATP. And we just talked about how 1 NADH will give up its pair of electrons in complex 1. And in so doing, 10 protons will pump across from the matrix to the intermembrane space. So 10 divided by 4, you get your 2.5 ATP. Now you could do a similar calculation for FADH2. But what you need to realize is that FADH2 does not start at complex 1. FADH2's pair of electrons is funneled from complex 2 onwards. So it means that for FADH2, it's really 4 plus 2, 6 protons. So let's do the same calculation. If 4 protons will give you 1 ATP, then the 6 protons, that is equivalent from 1 FADH2, will give you 1.5 ATP. So that's how we get the 2.5 and the 1.5. So please note that we just took a little piece of oxidative phosphorylation. This is not a full explanation of oxidative phosphorylation, but it's a very important piece. So let's recap. We said that in glycolysis, we got a net gain of 2 ATP. Link reaction, there was no ATP being formed there. In the Krebs cycle, we got 2 ATP. So let's look at oxidative phosphorylation. Now with the new numbers, 2.5 and 1.5, we get 28 ATP. So in total, that means that for each glucose molecule that is under aerobic respiration will give you as much as 32 ATP. So now we have the number 32. And then again, just to point out, alright, the bulk of ATPs that are being produced in aerobic respiration is coming from oxidative phosphorylation. You're getting 28 of the 32 ATP. But there's something else that we have to take into consideration. Let's go back to the link reaction, the TCA cycle, and glycolysis. In all three reactions, we are producing NADH. Alright, but we have to also take into consideration the location of these metabolic processes. Where are they taking part in the cell? Well, the link reaction in TCA cycle, they are taking part in the mitochondria. Alright, and we also know that oxidative phosphorylation takes part in the mitochondria. So the link reaction in TCA cycle, their NADH will go straight to oxidative phosphorylation. Easy peasy. But for glycolysis, that's taking part in the cytosol. So it means that the NADH being produced by glycolysis is being formed in the cytosol. It will mean that this NADH has to go to oxidative phosphorylation, it has to go into the mitochondria. But what we know is that there are no transporters on the inner membrane of the mitochondria for NADH. In other words, NADH can't go in and likewise it can't come out of the mitochondria. There are no transporters for NADH, and that's a good thing because you want all the NADH from link reaction in TCA cycle to go to oxidative phosphorylation. You do not want the mitochondria to be leaking out any NADH. So there are no transporters for it. So it means that if the NADH that is produced by glycolysis can't get into the mitochondria, they must have some way that at least to get a pair of electrons from the NADH molecule to oxidative phosphorylation. In other words, for it to reach the electron transport chain. Now, there are different shuttle systems that the mitochondria use to get that pair of electrons from the NADH into the electron transport chain. We're going to be looking at two of them. The first one we're going to be looking at is Glycerol 3-phosphate shuttle, also known as, in some textbooks, as Glycerol phosphate shuttle. Now let's look at where we are. So outside here, that's the cytosol, so that's where glycolysis will be taking place. And everything here represents the mitochondria. So you have the outer membrane, you have the space between the outer membrane and the inner membrane, which is the intermembrane space, the inner membrane, and then the matrix. So DHAP, which is dihydroxyacetone phosphate, reacts with NADH plus H plus to give you NAD plus and glycerol 3-phosphate. The glycerol 3-phosphate that is formed in the cytosol then enters the mitochondria. There's an enzyme on the surface of the inner membrane of the mitochondria that reacts with glycerol 3-phosphate and FAD to give you back your DHAP, and one molecule of FADH2 is produced. Now that one molecule of FADH2, we know from before, will produce 1.5 ATP. The DHAP that is made in the mitochondria returns to the cytosol. So that's the glycerol 3-phosphate shuttle system. But there's something to note here. You started with one NADH, alright, which we know produces 2.5 ATP, but we end up with one FADH2 which produces 1.5 ATP. It means that we are short of 1 ATP. Now, for glycolysis, it's 2 NADH, so it means that we're going to be short of 2 ATP. So it means that for under aerobic respiration using the glycerol 3-phosphate shuttle system, one glucose molecule will really give you 30 ATP. Now this shuttle system is used a lot by muscle, and it enables the muscle to sustain a very high rate of oxidative phosphorylation. So that's the glycerol 3-phosphate shuttle system. Let's look at another shuttle system called the malate-aspartate shuttle system. So once again, let's make sure we know where we are. This here is the cytosol, so that's outside of the mitochondria. And then this is the outer membrane of the mitochondria, the intermembrane space, inner membrane matrix. So all of this is the mitochondria here. So we start inside of the cytosol. So OAA, which is oxaloacetate, is reacting with NADH plus H plus to give you malate plus NAD plus. That's taking place in the cytosol. Now that malate, once formed in the cytosol, then enters the mitochondrial matrix. When it reaches the mitochondrial matrix, the malate plus NAD plus is converted to oxaloacetate plus NADH plus H plus. As we know before, the NADH could generate 2.5 ATP. So you started with NADH, and via this shuttle system, you end with NADH. So it means that for this malate-aspartate shuttle system, as opposed to the one that we saw before with glycerol 3-phosphate shuttle, this one, there's no loss of ATP. All right. Now the oxaloacetate undergoes two reactions, all right, and it then returns to the cytosol. So that's the malate-aspartate shuttle system. So for one molecule of glucose undergoing aerobic respiration using the malate-aspartate shuttle system will give you your 32 ATP. There's no loss of ATP there. So again, the number of ATP depends on the tissue and the shuttle system that it's operating. All right. Now the heart and the liver uses the malate-aspartate shuttle. Now let's look at anaerobic respiration. Now in anaerobic respiration, you're getting only 2 ATP. That's significantly less than what we saw we were getting with aerobic respiration, which was 32 ATP. But the answer to that is a simple one. For anaerobic respiration, there's no oxygen available, so therefore oxidative phosphorylation, which is the where you get the bulk of ATP from, is not happening. So let's look at what's happening with anaerobic respiration. Again, you're using glycolysis, and you're going to convert one glucose to 2 pyruvate molecules, and there you're going to get a net gain of 2 ATP, 2 NADH. Now those 2 pyruvate molecules, depending on the tissue and the organism, they will either produce lactate or ethanol via fermentation reactions. Now these fermentation reactions do not contribute to the ATP count. In other words, they don't produce any ATP, but they have a very important function. And that important function is that it's going to regenerate NAD+. So it converts the 2 NADH that you got from glycolysis, it's converting the 2 NADH to 2 NAD+, so that glycolysis could continue. All right? But in terms of ATP, these fermentation reactions do not add to the ATP count. So at the end of the day, via anaerobic respiration, one glucose molecule is going to produce 2 ATP molecules. So guys, I hope you liked the video. You learned a lot. I hope so. Like, comment, subscribe. Thanks for listening. See you later. Bye.